Assalamu alaikum. Inshallah, in this lecture, we'll be talking about recombinant proteins and how we can take advantage of them. So, clinically as well as in research. So, let's start. First, we'll be talking about the production of human proteins in bacteria. Okay. So, remember the things that we talked about before in terms of cloning, how we can clone DNA into a plasmid. But we will start talking about the production of complementary DNA or cDNA. Now, in order to express a specific gene in bacteria, a human gene that is, we have to select its messenger RNA. Now, there are two problems when it comes to using the messenger RNA, a human messenger RNA, for any particular gene. The first problem is that there are so many RNA molecules inside the cell. Now, I told you before that like 50 years ago, um, about three RNA molecules were known, and these were messenger RNA, tRNA, and ribosomal RNA. Then other molecules, other RNA molecules, since then have been discovered. Now, so far we have like 15, 20 different types of RNA molecules, and there are so many of them in terms of number of molecules inside cells. So we have the messenger RNA, we have the classical ribosomal RNA and tRNA, but we have other types of non-coding RNA, that is RNA molecules that, again, that are transcribed but not translated. And they can be small and long. We talked about the long non-coding RNA molecule, long non-coding RNA molecule. Now, I did mention circular RNA briefly. Nothing much to talk about so far in terms of circular RNA, its nature and what it does exactly. Now, we have the small non-coding RNA molecules. We have the microRNA. Okay, we have the microRNA. We talked about the microRNA in our previous lecture. So we have all of these RNA molecules. Now, how can we select messenger RNA molecules specifically? Well, messenger RNA molecules are probably the only ones that have a poly A tail. All of the other RNA molecules do not have a poly A tail. So what we do is that in order to convert RNA to DNA, and then we can use this DNA molecule as a gene to express a human protein in bacterial cells, we use reverse transcriptase, which it's the vital enzyme that converts RNA to DNA. And this enzyme requires a primer, just like DNA polymerase does. So we use a poly T primer. Okay, so we add this poly T primer, and the reverse transcriptase synthesizes the first strand of DNA. And that's the solution. This is how we can select, this is how we can selectively use messenger RNA, but not all of the other RNA molecules. Is that we use the poly T primer again for reverse transcriptase to synthesize the first DNA strand. Okay, and that's the solution. Problem number two is that we have introns in genes. So if I want to synthesize DNA from DNA, if I want to synthesize a gene starting with the genomic DNA, I'm gonna have a problem with introns. So, and this is the reason why we use mature messenger RNA, meaning that a messenger RNA that is polyadenylated. So what we do is that we don't use DNA to make copies of a gene because of the presence of introns. We do not use the primary messenger RNA that contains introns and exons. Rather, we use mature messenger RNA um, that contains only exons connected to each other without any introns. And what we do, again, we use reverse transcriptase, that's the solution, to synthesize the DNA from the messenger RNA, and this messenger RNA is already spliced. So we make, again, double-stranded DNA, which is known as the complementary DNA. So now we have solved two problems. The problem number one is how we select messenger RNA and disregard all of the other types of RNA molecules. And the second problem is how we can get rid of introns. Well, we use mature messenger RNA that is already spliced. So now we have a complementary DNA that is basically a gene that contains only the exonic parts, only the coding parts. We take this complementary DNA and then we can clone it into a plasmid. Now, the whole purpose, again, is to synthesize human proteins inside bacteria. Remember, that's bacteria do not have introns and exons. Bacteria do not have their messenger RNA molecules do not have poly A. So, but now we have a cDNA that represents a human gene without introns and without the poly A, by the way, because we can remove it, we can get rid of it. So we clone it. So basically what we do is that to express a human gene inside bacteria, we use an expression vector. Well, the vectors that we used before for cloning genes or for cloning DNA, these are called cloning vectors. The whole purpose of cloning vectors is just to amplify DNA or to have copies of DNA amplified inside bacterial cells. Regardless of what this DNA is, it can represent introns, exons, promoters, enhancers, you name it. But here we want to express a human protein, starting with human messenger RNA that is already spliced. So we convert the messenger RNA to cDNA, and then we clone the we clone the complementary DNA into an expression vector. Now this expression vector has the same components as cloning vectors: the origin of replication, a cloning site, and an antibiotic resistance gene that would allow us to select bacteria that contain this plasmid. But expression vectors have three additional components. Component number one is a promoter sequence, a promoter that bacteria can handle. Now remember, it doesn't have to be the same promoter as the original gene, okay? So like insulin gene, for the human insulin gene, I don't have to use the insulin gene promoter to express the insulin gene inside bacteria. I can use bacterial promoters. Molecules are stupid. I can fool them. So I'm going to have a promoter that functions, that works. And then I need a cloning site, of course, where I can place that gene downstream of the promoter. So I'm going to have the promoter right here. Downstream of the promoter, I can clone the gene. Okay. And then once I express the gene, in order to translate the messenger RNA in bacteria, we need a Shine-Dalgarno sequence. This is the ribosomal binding sequence. This is the sequence where the small ribosomal subunit binds to. Remember that? So go back to the transcription lecture. So I need this sequence, the Shine-Dalgarno sequence, the SD right here. Okay, I need that. And remember, we need a transcription termination sequence, not a stop codon. I'm not talking about that. We need a transcription termination sequence so that the RNA polymerase that synthesizes the messenger RNA knows that it has to stop mRNA synthesis right there. So we can clone the human gene right downstream of the promoter. The Shine-Dalgarno sequence would be part of the messenger RNA, but then I have an AUG start codon right here, and we're going to have a termination sequence downstream of where the gene is cloned, so that transcription is terminated. And of course, now the cDNA, well, we can have a stop codon right here as well, so that we would have a termination of translation as well. Now, question for you though, question for you. Um, and I think I asked you this question before. Will the cDNA contain the stop codon for translation or not? Well, regardless, eventually what we're going to have is the following. So we can clone the plasmid, or we can transform bacterial cells with the plasmid. That is, we insert the plasmid into bacterial cells, and bacterial cells are going to recognize the promoter region, and they're going to start RNA synthesis or messenger RNA synthesis, transcription of the messenger RNA, and then they're gonna translate the messenger RNA into a protein. So we're gonna have the human protein expressed inside bacterial cells. Now, again, cells are stupid. We can fool them. Bacteria thinks. We use a bacterial promoter, by the way. Okay, not the insulin gene promoter, for example. So bacteria can be fooled so that they would transcribe the gene of interest, this one right here, starting with their promoter region. And they're gonna, in transcription, they're gonna translate the messenger RNA into a protein, starting with because there's a Shine-Dalgarno sequence and so on. And there's stop codon, start codon, stop codon, same thing as what bacteria is used to. And protein can be folded. So actually, this has worked really well, because now we can produce, by the way, the human insulin protein inside bacteria, growth hormone, plasminogen activator for blood clotting, and so on. So we can we convert bacterial cells into factories that can produce human proteins, and these proteins are used clinically to treat patients. If I take the bacterial cells and I open them up and I isolate, purify, extract the proteins, so I isolate the proteins away from the DNA, RNA, and other molecules, and I do SDS page, sodium dodecyl sulfate, which is the detergent that denatures proteins, polyacrylamide gel electrophoresis, we can isolate proteins based on size. Okay, so you can go back to the biochemistry lecture. So here we have all bacterial proteins, and this is taken from bacteria. These proteins are taken from bacteria that do not contain the plasmid. And here we have the bacteria that contain the vector, the expression vector. And you can see the only difference is this band right here. And this band represents the human protein. So they express huge amount of human protein. Okay, so this is expression vectors. Now the problem with expressing human proteins in bacteria is that bacterial proteins do not have disulfide bonds like human proteins. That's a problem. Bacteria do not have post-translational modification. There's no glycosylation of proteins, for example. Proteins can be misfolded, especially large proteins. So bacteria cannot handle large human proteins, but they can handle small proteins. And sometimes bacteria would degrade human proteins. They would say, what is that? I cannot recognize this protein. It looks funny. It looks awkward. It looks different, so they can degrade it. Now, as a solution, we can use yeast. Yeast cells are, single cell organisms. They are eukaryotic systems, so they do what human cells do in general. So, and they grow fast. Okay, and we can insert plasmids inside them. Again, cells are stupid. They look at promoters. They don't know this is plasmid or genomic DNA. They can do exactly what we want them, what we ask them to do, or what we force them to do. We can fool them again. So we can use yeast to produce human proteins because they can handle human proteins better than bacterial proteins. So this is expression of human proteins in bacteria. One thing we can do is that we can create a recombinant protein. Remember, recombinant means, if I say recombinant plasmid, it means that it's a plasmid that contains a non-bacterial gene or piece of DNA or something different than what a plasmid contains. A recombinant protein is a protein that contains two different proteins, for example, or different parts of proteins. So we can create a recombinant protein inside bacteria or yeast or even human cells. And what we do is that, for example, we take gene A, gene B. Now gene A will produce protein A, gene B will produce protein B. We can put them together as one gene. So, for example, we delete the stop codon right here. And we put the genes together so that when they are translated, they're translated, when the protein is translated, when the protein is synthesized, synthesis of the protein does not stop here at the stop codon. So the synthesis of the polypeptide continues. So you have both parts, both proteins expressed, synthesized, and then you have each protein fold by itself, independently of the second protein. Now, there's a linker right here, okay? There's something that links the two proteins together. And this linker does not really affect the protein folding. So we're gonna have a recombinant protein, protein A and protein B produced together. And we can do this with, we can do different combinations of proteins, by the way. And I'll give you an example. There is a protein known as green fluorescent protein. It's a protein that was isolated from jellyfish. Remember, if you see these documentaries on the Sea World, there are these jellyfish that can produce fluorescence in their legs. Now, one of these proteins is known as a green fluorescent protein, and it gives a green fluorescence. This is how it looks like. So what we do is that we can take the green fluorescent protein gene, and we can clone it upstream or downstream of another gene. So when you have synthesis, we can put it in an expression vector, and you have the production of a messenger RNA, and this messenger RNA is then translated into a protein that is large. It contains the green fluorescent protein, and it contains the protein of interest. Now, this protein right here would fluoresce because this protein, the green fluorescent protein, fluoresces as well. Okay, so let me try to play this. Okay, okay, that's jellyfish, right? Beautiful movie. You can see the fluorescent proteins moving, right? So the gene that produces these proteins have been isolated. One of them is the green fluorescent protein, and we can again clone the, the genes together. So the green fluorescent protein would become part of the protein of interest, and each one of them folds independently of the other one. Okay, that's the advantage of domains, right? We go back to the definition of a domain. So this protein right here fluoresces. Okay, so wherever this protein goes inside the cell, I can see where it's going. We can, see the cell live, by the way. So, so these are some examples of proteins that that have been tagged. Tagged means that we attach another protein to them or a piece of proteins or whatever. This is a whole cell. These are different cells that express the green fluorescent protein, and the whole cell fluoresces. This is the actin protein with the green fluorescent protein. So you can see the actin filaments fluorescing. These are, here we have mitochondrial proteins or a mitochondrial protein tagged with the green fluorescent protein. So the green fluorescent protein is part of the mitochondrial protein, so the whole mitochondria can fluoresce. This is the tubulin protein, the alpha tubulin protein. So you can see the microtubules right here. You can see how they are organized. We can actually have organisms like fruit flies fluorescing, or rabbits or mice fluorescing. We can see tissues, by the way. This is a whole fish. So we can also have nerve cells fluorescing by letting them express the green fluorescent protein. And we can see how these nerve cells can connect with each other. So we can understand nerve connection, for example. We can understand the morphology, the structure, the shape, the organization of tissues and organs, and even whole, organisms. Anyhow, so these are some examples, just to show you the things that we can do with the green fluorescent protein. Now, let's go into something similar but different at the same time. How can we understand protein-protein interaction? Now, I'm going to introduce two techniques to you. One of them is known as co-immunoprecipitation. Precipitation means, it settles down. بترسب, ترسيب. Immuno, once you hear the word immuno, it means I'm using antibodies here. Co means together. Okay, so I'm precipitating a protein with another protein that is accompanied by another protein using antibodies. Remember that we can have, let's say, a large protein. Let's say this protein MLL has two domains, an N-terminal domain and a C-terminal domain, and the N-terminal domain can interact with these two different proteins. And these two proteins can also interact with each other. Now, the C-terminal domain can interact with 4 proteins. Some of them can interact with each other and others cannot, like this protein right here cannot interact directly with this protein, and so on. Well, how can I identify and discover the protein-protein interaction. So with the immunoprecipitation, that's what we do. We have these protein complexes. A protein complex is when you have multiple proteins interacting with each other. What we do is that we add an antibody, and this antibody is specific to one single protein. Now, this antibody then targets this protein, the pink protein right here, and the pink protein interacts with other proteins. So what's going to happen is that this is really quite heavy, and it's going to precipitate. It's going to go down. And we remove everything else, everything that does not interact with the antibody would be removed. And then we have these proteins right here, or the protein complexes, I'm sorry, that interact with the pink protein. We can take this complex and we can study it using, for example, STS page, gel electrophoresis, or using Western blots. Remember, Western blots are immunoblots, where we separate proteins based on size, and then we transfer it to a membrane, and then we add a primary antibody and a secondary antibody to detect the presence of a protein, and we can also determine the size of that protein. Okay, so you can go back to the biochemistry lectures again. Okay, so now by precipitating, by isolating this pink protein, we can identify all of the other proteins that can interact with that pink protein using, again, immunoblotting. That's immunoprecipitation. Let's go into something a bit more sophisticated. Okay, it's known as the yeast to hybrid system. So it's a genetic system where we express hybrids of two proteins or two genes. So first, this is, I'll give you an introduction. There is a genetic system. There is sort of like a regulatory sequence called UAS. Okay, it's called UAS. This is like a promoter proximal element, you name it. It's the binding site of a transcription factor. This transcription factor is known as the GAL4. GAL4. This GAL4 protein has two domains, a DNA binding domain and an activation domain. Okay, let's go back to the definition of a domain. A domain is a three-dimensional structure of a protein, and it can fold independently of the rest of the protein, and it maintains its function. So we have two domains, DNA binding domain and an activation domain. This activation domain can activate the expression of a gene. Except that the gene that we use, see, that's the power of genetic engineering. The gene that we use is a reporter gene. Just like luciferase, luciferase is a reporter gene. It's a gene that gives us a piece of information. It reports something. It's a reporter. The gene that we use for the yeast to hybrid system is the LAC-Z gene. It's the beta-galactosidase gene from bacteria. It's the gene that produces galactosidase, which is an enzyme that cleaves lactose. Okay, so we use a regulatory element from yeast, and we put it in a DNA, upstream of a reporter gene, and the reporter gene is the bacterial beta-galactosidase gene that cleaves lactose. Now, okay, got it so far? So, if LacZ is expressed, if beta-galactosidase protein is produced, then it can cleave lactose, except that we use a substrate that is similar to lactose, and it's called X-gal. So it looks like lactose, except that beta-galactosidase would cleave X-gal, producing this molecule right here, which gives a blue color. So the yeast cells become bluish. Okay, got it? So we have this genetic system. Again, the reporter gene is now under control of the UAS genetic system. The UAS system or element is the binding site of the GAL4 transcription factor, which has adenine binding domain and an activation domain. If GAL4 binds to UAS, then we have expression of the reporter gene by the RNA polymerase, and then translation of the messenger RNA into beta-galactosidase. If we add X-gal to these yeast cells can take up X-gal, they can cleave it, and they can produce this substrate that gives a blue color. Now remember that if we grow yeast on a plate, on a dish, each cell can form a colony, So right here we have different colonies. Some of them are white and others are blue. The bluish colonies are the yeast cells that express the reporter gene. And the white yeast colonies originate from a yeast cell that does not express the beta-galactosidase. Got it so far? Okay, so let's, can we zoom in? No, we cannot zoom in. Okay, so that's the idea. So we have these white colonies that do not express LAC-Z gene or do not produce the beta-galactosidase, and we have the blue colonies that produce the beta-galactosidase. Okay, so what do we do? Remember what the definition of a domain is. So we create a recombinant protein. And the recombinant protein is the following. So we have gene A that has 2 domains, and we have gene B that has 2 domains. Well, we can create a recombinant gene that has the portion that produces this domain from gene A and this domain from gene B, and we can put them together. So we produce a recombinant protein that has 2 different domains from 2 different proteins. Remember that this domain maintains its function, and this domain maintains its function as well. So remember that. So how can we, what's the purpose of the yeast to hybrid system? To test if protein A can interact with protein B. How do we do that? Well, we use the genetic system that I just told you about. The UAS with the GAL4, which has 2 domains, and we have the reporter gene, the LAC-Z, and we add to the yeast cells the X-gal. So what do we do? Now, we want to test if protein X interacts with protein Y. We're going to use the same genetic system that we just talked about. The UAS regulatory element, GAL4, and the LAC-Z gene that produces the beta-galactosidase. And that's what we do. We create 2 plasmids. And these 2 plasmids, they are expression plasmids. We're going to put on one of them, gene X, the gene for protein X. And it would be produced as a recombinant protein with the Gal4 DNA binding domain. So when X is produced, it's produced with the Gal4 DNA binding domain. And we have plasmid 2. And this second plasmid contains the gene for protein Y. And it's produced as a recombinant protein along with the Gal4 activation domain. So we're going to have protein Y with the activation domain of Gal4. The idea is that if X interacts with Y, then these two would be close to each other. So we're going to have, again, this protein of interest with the DNA binding domain. And we're going to have this another protein of interest, protein Y, with the activation domain. Now, if these two interact with each other, then we're going to have the DNA binding domain and the activation domain very close to each other, and we're going to have transcription of flag Z. If flag Z is expressed, we're going to have beta-galactosidase. If beta-galactosidase is expressed, if it's there, it's going to cleave X-gal. If X-gal is produced, is cleaved, it's going to generate a a blue product. And the yeast cell and the yeast colony becomes bluish. And the color blue indicates that X interacts with Y. If X does not interact with Y, then the DNA binding domain would not be close to the activation domain, and as a result, there would be no transcription of the LAC-Z gene, there would be no production of beta-galactosidase, there would be no cleavage of X-gal, there would be no blue product, and the colony would look whitish, not bluish. Got it? So we have these two probabilities. Either X interacts with Y, a blue colony, X does not interact with Y, white colony. White colony. So the previous example was simple. I'm dealing with X and Y, just one single protein, X, and one single protein, Y. Now, let's make things a bit harder. I want to identify all of the proteins that can interact with X. Not a single protein. I'm not testing a single protein. I want to identify all of the proteins that can interact with X. So, we create a cDNA library. Remember what it means? It's a collection of plasmids, and each plasmid contains a cDNA representing a specific gene. That's a cDNA library, a collection of different plasmids. So I have plasmid, this plasmid right here, producing protein A. It contains the gene for protein A. And this plasmid has gene B for protein B. This is protein C. This produces protein C. This produces protein D, and so on. This is a cDNA library. So I have different plasmids that can produce proteins, recombinant proteins. Each protein is produced with the activation domain of Gal4. And I have one single plasmid that can produce protein X, and this protein X is produced as a recombinant protein with the DNA binding domain of Gal4. So we take this and we introduce it into yeast cells. So this plasmid right here that produces recombinant X, would exist in every yeast cell. And I have, let's say, 1 million yeast cells. Each one of them has the plasmid for the recombinant X gene, or that can produce a recombinant X protein with the DNA binding domain. And then I take these 1 million yeast cells and I insert these plasmids into these yeast cells. So each yeast cell would take up one of these plasmids. Okay? So each yeast cell would have two plasmids. The plasmid that produces protein X, recombinant protein X, and one and a plasmid that would produce either recombinant protein A or recombinant protein B or recombinant protein C or recombinant protein D. Okay, so each yeast cell would have two plasmids. Now, and we grow them on a plate. If X interacts with A, then this yeast cell would have the production of the beta-galactosidase that can cleave X-gal into a bluish product, and the colony would look blue. If in this yeast cell X does not interact with protein B, it means that the binding domain and the activation domain would be far away from each other. They wouldn't interact with each other. They wouldn't be close to each other. And as a result, there would be no production of beta-galactosidase. There would be no cleavage of X-gal into a blue product, and the colony would look white. So we can have a plate that has a lot of yeast colonies. Some are white, some are blue. And I'm interested in the blue colonies. Okay, so what we do is we take these blue colonies and we isolate the plasmids and we see what the identity of the gene that is contained in these plasmids. Now, each yeast cell or colony would have one of these plasmids. So what we do is that we take these plasmids, okay, we grow the yeast cells, we isolate the plasmids, and we see what the identity of the gene that is included, that is cloned into this plasmid is. And we can identify what the protein is or what the gene is. And this is the gene that can produce a protein that interacts with X. So we can identify the X partner. All right? So these are the probabilities now. If we have a plasmid that produces the full Gal4 protein, this is the binding domain, the binding domain, and the activation domain, then LAC-Z would be expressed. Beta-galactosidase would be produced. And if we add X-gal to these yeast cells, we're going to have a blue colony, right? Because we have the whole Gal4 protein. If we have a plasmid that produces the DNA binding domain only, then there would be no production. If we have a plasmid that produces the activation domain only, we're going to have no production of beta-galactosidase, okay, because we need the DNA binding domain in here. But if we produce protein X, protein Y, and these two proteins can interact with each other with the DNA binding domain being recombined, being part of protein X, and the activation domain being part of protein Y, then these two would be close to each other. The activation domain, the binding domain, we're going to have expression of LAC-Z, production of beta-galactosidase, conversion of X-gal into a blue product, and the colony would look blue. We take these cells, this colony, we isolate the plasmid that contains this gene, because I know protein X or gene X. I want to identify this gene or protein that can interact with protein X. So we isolate the plasmid, and we can do sequencing, we can do immunoblots, whatever, and we can identify this unknown gene. Okay. So again, I'm interested in the colonies that are blue. These are the colonies where we have yeast cells that produce a protein that can interact with protein X. So I'm going to leave you with this slide. This is taken from the book. It contains a lot of information. Okay, it explains the technique that I just told you about, and you can read it on your own. Okay, and Allah ma'akum.